In this graph, we're going to look at carrier transport rates and the factors that affect it. On our y-axis over here, we're going to graph the rate of solute transport across the membrane relative to, on our x-axis, the concentration of solute. In other words, the density of the solute adjacent to those transport proteins. Now, let's contrast carrier-mediated transport relative to channel transport before we move on. Carriers interact directly with the solutes that they move across the membrane. This is not true of channels. Channels do not directly interact with the solutes that pass through them. This allows channels to move solute very, very rapidly across membranes, 100,000-fold faster than carrier-mediated transport. The downside of channel transport is that it is always passive. It always only allows solutes to move down their concentration gradients. This could be true of carrier-mediated transport as well, facilitated diffusion, but we could also have scenarios where we are driving solutes against their gradients using ATP or the energy dissipated through the gradient of another solute, co- or countertransport. Now back to our graph here. We can see as we increase the concentration of solute moving to the right here on our x-axis, what happens is when we have a low concentration of solute, shown here, we have a reduced likelihood of interaction with our carrier protein, and our rate of transport across the membrane would be relatively low. Right, down here. If we increase the concentration of the solute in the vicinity of these transporters, we then, of course, have moved to the right, and we have increased the likelihood of interaction with our transport proteins, and our transport rate has increased. Okay, so our concentration of solute is directly affecting our rate, our ability to move solute. We could also change the number of transport proteins. We have two different lines here. If we have fewer transport proteins, we're on the red line. If we have more transport proteins, we're on this blue line. At a given concentration, if we have more transport proteins, if I add another transport protein over here, we increase the likelihood of interaction with those transporters, and therefore our rate of transport would be higher. Finally, because carrier proteins interact directly with a solute, they have the ability to become saturated. This means that all of the carriers are bound to or moving solute at a given moment. Once that happens, we reach the transport maximum. Right? And so at that point, adding more solute, increasing the concentration, would no longer increase the rate at which we're moving that solute. They're all moving solute maximally. Of course, if we add more transport proteins by increasing their expression, we have the ability to increase our transport maximum. You can also see that our transport maximum is delayed, right, if we increase that number of transporters, because again, it would require more solute to reach that level at which all of the transporters are saturated with that solute.